Welcome to the deep dive. Have you ever wondered about, natural ways to approach those really stubborn health issues, like pain and inflammation, especially the chronic kind?
Absolutely.
It feels like you can get totally lost trying to figure it all out, wading through tons of complicated information.
It's definitely a challenge. There's just so much out there, and really understanding the science behind natural compounds, well, it takes some careful digging into the research.
Yeah, that's exactly why we're here. Today, we're doing a deep dive, really getting into a stack of sources someone sent us. They were really curious about this molecule called palmitoylethanolamide. You'll probably see it shortened to PEA.
Right, PEA. It comes up quite a bit.
So our mission today is to sift through this whole research bundle, and it's a real mix. Scientific reviews, some specific animal and cell studies, clinical trial info, basically. Pull out the key insights for you. Cut through that complexity and figure out what the science actually says about this natural lipid.
And this collection looks pretty good. It gives us a nice range. We can look at the basic biology, how PEA works, then examine findings from, experimental models, and also see what's popping up in human studies. We'll be using that document, the Palmitoylethanolamide in-depth research request, kind of as our main guide, but pulling in details from all the related sources too.
Perfect. All right, let's unpack this molecule then. Palmitoylethanolamide. Where do we even begin? What is this thing, really?
Okay, so at its core, and this is fundamental across all these sources, PZA is a naturally occurring lipid signaling molecule.
Lipid signaling. Oh.
Yeah, it belongs to a family called N-acetylethanolamines.
N-acetylethanols, got it. So when you say naturally occurring, you mean it's something our own body actually makes, like an internal signal, a messenger our cells use.
Exactly, yes. It's produced endogenously. That means it's synthesized right inside your own cells, your own tissues, usually when it's needed. The sources really hammer this home. It's part of your body's own communication system. Now, you can find it in small amounts in certain foods, like egg yolks or soybeans. But the main focus of all this research is really its role as something your body makes and uses itself.
That's really interesting. So our body has this built-in system using molecules like PEA. Okay, but how does it work? Once it's made, how does this molecule actually do anything? How does it exert its influence, according to the science in these sources?
Right. This gets us to the cornerstone mechanism. It's discussed quite a bit, especially in that in-depth research request guide we're using. The main way PEA seems to work, based on all this research, is by interacting with a very specific type of receptor. It's called peroxisome proliferator activated receptor alpha.
Peroxisome proliferator. Okay. Let's just call it PPAR alpha. That's a mouthful.
PPAR alpha, yes. Much easier.
So PPAR alpha, what kind of receptor is that? Is it sitting on the outside of cells waiting for a signal like a lot of receptors we hear about?
No, actually, and that's a really key difference. PPAR alpha is a nuclear receptor.
Nuclear. So
Yeah.
inside the cell nucleus, where the DNA is.
Exactly. It's located right inside the nucleus. And the process described is, well, it's pretty neat. When PEA gets into the nucleus and binds to PPAR alpha, they don't just act alone, they actually team up. They form this partnership. It's called a heterodimer.
Heterodimer. Okay, a two-part team?
Yeah, with another receptor called the 9-cis-retinoic acid receptor, or RXR.
So PEA meets PPAR alpha inside the nucleus. They grab RXR, form this team.
Right.
Then what? What does the team do?
So this new complex, this PPAR alpha XR team, it then goes and finds specific spots on the DNA. These are in the promoter regions of certain genes, kind of like the on switches or control panels for those genes.
Oh.
These specific DNA sequences are called PPAR response elements, or PPREs.
PPREs.
Yeah.
Got it. Specific landing pads on the DNA.
Precisely. And by binding onto these PPREs, this whole complex can then modulate, basically turn up or turn down the expression of those specific target genes.
Right, like a dimmer switch almost.
That's a great analogy. Yeah, it controls how much protein those genes produce, and that in turn changes how the cell behaves. That in-depth research request guide even gives a measure of how strongly they interact. It notes PEA activates PPIR alpha with something called an EC50 of about 3 min.
Okay, hang on. EC50, 3 NO, or micromolar. What did that actually mean in terms of, how potent this is? Is 3 NM a lot or a little?
Right, good question. So the EC50 tells you the concentration of PEA needed to get half of the maximum possible activation of that PPAR alpha receptor in, a lab test. So 3 micromolar is the concentration for 50% effect.
Okay.
Now, translating that directly to what's happening in every tissue in the body is tricky because local concentrations vary, but it gives scientists a standard benchmark. It tells them PEA has a pretty significant measurable effect on this receptor at these micromolar levels. It's not incredibly potent compared to some drugs maybe, but it's definitely potent enough to be biologically relevant.
Understood. So, backing up to the mechanism, it's like PEA is a key that gets inside the nucleus, teams up with these other molecules, finds the right spot on the DNA, and then fine-tunes these genetic switches, directly changing how the cell functions. That feels like a very fundamental way to affect biology.
It absolutely is. It's influencing things right at the genetic level.
Yeah.
And the sources are quick to point out where this PPAR alpha mechanism is particularly important. Yes, PPAR alpha is highly expressed in tissues involved in, say, fatty acid metabolism, liver, heart, skeletal muscle.
Right, metabolic stuff.
But crucially, it's also found in immune cells and in specific areas of the brain.
Ah, okay. Immune cells and the brain. That sounds important for the effects we're talking about, like inflammation and pain.
Exactly. That distribution is absolutely key to understanding why PEA seems to have these effects on inflammation, marrow inflammation, and pain signaling that the research discusses. The switch has to be present where the action is needed.
Makes total sense.
And the sources really emphasize how critical this PPAR alpha activation is. They cite experimental studies, for example. There's the spinal cord injury study mentioned. I think it was published in the Journal of Neuroinflammation, where if you block PPAR alpha using drugs, or if you use animals genetically engineered to lack PPAR alpha, then the protective effects you normally see with PEA in those injury models are drastically reduced, or sometimes they just disappear altogether.
Wow. So that's strong evidence that this pathway, this PPAR alpha switch, is really doing the heavy lifting for PEA's benefits in those specific situations.
It provides really compelling evidence, yes. It tells us that pathway is essential for many of the effects being reported in these experimental models.
Okay, so that PPAR alpha pathway is clearly central, but, biology is really just one simple pathway, right?
Yeah.
Are there any other ways the sources suggest PEA might be working or contributing?
You're absolutely right to ask that. Biology is complex, and while PPAR alpha is definitely the star player in these sources, they do explore some other possibilities or related mechanisms.
Like what?
Well, for instance, that same spinal cord injury study we just mentioned, it actually found that PEA's positive effects in that particular model weren't only dependent on PPAR alpha. Yeah, they found that PPAR delta and PPAR gamma receptors, other members of the same PPAR family, were also involved. When those were blocked or missing, PEA wasn't as effective.
So other related genetic switches might play a role too, at least in some contexts.
It seems so in that specific model. And interestingly, that study also suggested PEA might even help restore the levels, the expression of those PPAR delta and gamma receptors in the injured spinal cord tissue. So it hints at some really complex regulatory feedback loops going on.
Huh, that's fascinating. What else? Any other potential mechanisms mentioned?
Another idea that comes up in a couple of the sources is this concept of an entourage effect.
Entourage effect. I've heard that mainly in relation to cannabis compounds. How does it apply here?
Right, it's a similar idea. The suggestion here is that PEA might potentiate, basically boost or enhance the effects of other related molecules your body makes, specifically other N-acetylethanolamines like anandamide or AEA.
Anandamide, yeah, the bliss molecule, one of the main endocannabinoids.
Exactly. So the idea mentioned in relation to some gut inflammation research and studies on blood vessels is that PEA might make anandamide work better or last longer. This enhancement might involve interactions with receptors like TRPV1, which is involved in pain and heat sensation, or maybe even CB1, the main cannabinoid receptor, even though PEA itself generally isn't thought to bind strongly to CB1 or CB2 directly.
So PEA isn't necessarily fighting the main cannabinoid switches itself, but maybe it's acting like a volume knob for other molecules like anandamide that do, helping them send a stronger signal.
That's a good way to think about the entourage idea discussed here.
Okay.
It's potentially boosting the signal of its molecular relatives. And this kind of connects to another mechanism that's touched upon, an enzyme called NABA. Yeah, N-acetylethanolamine hydrolyzing acid amidosose. It's the main enzyme that breaks down PEA.
Okay, so it recycles PEA.
Right, but it also breaks down other N-acetylethanolamines, including anandamide, in some cellular compartments. Several sources mention NABA, including one perspective piece focused just on it.
Okay.
Now, while PEA is a substrate for NABA, meaning NABA breaks it down, there's research using NABA inhibitors, drugs that block NABA. And these studies show that blocking NABA can increase the body's own PA levels, for example, in models of colitis, gut inflammation.
So stopping the breakdown boosts the levels. Makes sense.
But it gets even more interesting. Some research suggests that PEA itself might actually influence the levels of other endocannabinoids like anandamide, AEA, 2-AG, and OEA. potentially by somehow affecting NAA activity or other related metabolic pathways.
Oh, okay, hold on. So PEA might act directly through PPAR alpha,
Mm-hmm.
maybe involve other PPARs, maybe
Oh.
boost related signals like anandamide through an entourage effect,
Yeah.
and it might influence the enzyme that breaks down itself and those related signals.
That's the picture that emerges from piecing together these different lines of research mentioned in the sources. It suggests PEA could be acting on multiple levels.
It sounds incredibly versatile, like a master modulator within this whole lipid signaling network in the body.
It certainly seems that way. It's clear from these sources that its effects are likely the result of several potential interactions. But again, that PPA alpha pathway stands out as the most consistently highlighted and strongly supported mechanism for the main benefits people are studying.
Okay, that gives us a really solid, albeit complex, understanding of how the science suggests this molecule works. Now let's get to the payoff. The so what? What are the actual effects the sources say PEA has on the body? Where does the research really show it helping?
Right, and based on the evidence presented in these sources, the effects are definitely strongest and most consistent when it comes to inflammation and pain.
Inflammation and pain.
Okay. That in-depth research request document is pretty direct. It states that PEA's activation of PPAR alpha kicks off this whole cascade of events leading to, and I quote, potent anti-inflammatory outcomes.
Potent anti-inflammatory outcomes.
Yeah.
That definitely sounds significant. How does that actually play out in the research? What specific anti-inflammatory actions are they talking about?
Well, the sources detail quite a range of actions down at the cellular and molecular level. So activating PPAR alpha leads to less production of those key pro-inflammatory signal molecules. The cytokines you mentioned earlier, things like TNF alpha, IL-1 beta, and IL-6. Several papers highlight this.
Okay, so it quiets down the main inflammatory messengers.
Exactly. It also inhibits the degranulation of mast cells.
Mast cells.
Yeah.
Those are the ones involved in allergies, right? But also inflammation.
Precisely. They're important immune cells that release histamine and other inflammatory stuff when activated. PEA helps stop them from spilling their contents. This is emphasized in several sources.
Okay, what else?
The sources also point to decreased activation of microglia.
Microglia. Those are the main immune cells in the brain, right?
That's right, and their overactivation is a huge driver of neuroinflammation, which is implicated in so many brain conditions. PEA seems to help dampen that down.
So it's not just calming inflammation in the body, but specifically in the brain, too, by affecting those key immune cells there.
It appears so. The research also mentions it helps attenuate oxidative stress.
Oxidative stress, that's damage from those unstable molecules, free radicals or ROS.
Exactly, reactive oxygen species. PCA helps reduce that damage. It also seems to reduce the infiltration of neutrophils, another type of immune cell that kind of rushes to the scene of inflammation. PEA helps limit how many show up.
Okay, fewer inflammatory cells arriving.
Right. And furthermore, they note decreased expression and activity of important inflammatory enzymes like COX2.
COX2, like what some anti-inflammatory drugs target.
Yes, exactly. And another enzyme called INOS, inducible nitric oxide synthase, which produces nitric oxide involved in inflammation. And related to that, it seems to inhibit nitric oxide production in macrophages, yet another type of immune cell.
Wow. It sounds like it's hitting the inflammatory process from multiple angles, reducing signals, calming down key immune cells like mast cells and microglia, reducing damaging oxidative stress, limiting immune cell traffic, and even dialing down inflammatory enzymes.
That's a great summary. It really does seem to modulate many different facets of the inflammatory response.
So where do we actually see these anti-inflammatory effects demonstrated in the studies mentioned? What kind of models or conditions?
The sources give several concrete examples. We talked about that spinal cord injury study. It showed less inflammation and less tissue damage in the mice treated with PEA.
Okay.
Then there are experimental models of intestinal inflammation, like colitis. Studies there showed PEA led to improved outcomes, like reduced neutrophil infiltration into the gut lining. And it also seemed to affect gut motility, how the intestines move.
Interesting. So effects in the gut, too.
Yes, and one source gets really specific, linking PEA's benefits in colon inflammation to its effects on enteric glia. These are support cells within the gut's own nervous system, and it involved a pathway using TLR4 and PPAR alpha. TLR4 is a receptor that recognizes bacterial components and triggers inflammation, so PEA seems to be intervening in that process via PPAR alpha in those gut support cells.
That's incredibly specific. Enteric glia. Wow.
Isn't it? Another study highlighted PEA helping regulate intestinal injury caused by radiation, and that effect seemed dependent on mast cells.
So different mechanisms depending on the type of injury or inflammation.
It seems plausible, and here's another interesting one. Even when PEA was given centrally injected directly into the brain in an animal model, it was shown to reduce inflammation happening out in the periphery, specifically in a paw edema model.
Wait, giving it in the brain reduced swelling in the paw. How does that work?
It suggests PEA acting within the central nervous system can influence inflammatory responses elsewhere in the body, likely through complex nerve signaling pathways controlling inflammation. It highlights a brain-body connection in regulating inflammation that PEA can tap into.
That is remarkable. Such a wide range of effects: spinal cord, gut, radiation injury, even central control over peripheral inflammation.
It definitely paints a picture of a broad anti-inflammatory potential across different systems, and very closely related to this, of course, is pain relief, analgesia.
Right, pain and inflammation often go hand in hand.
They do, and the in-depth research request document is particularly strong on this point. It explicitly states that the evidence for PEA's benefits is, quote, most robust when it comes to managing chronic and neuropathic pain.
Most robust. That's strong language in science.
It is, and it backs this up by mentioning there are multiple meta-analyses of randomized controlled trials supporting this.
Multiple meta-analyses. Okay, that's usually considered high-level evidence, summarizing results across many studies. Why is the evidence particularly strong for chronic and neuropathic pain, do the sources say?
Well, think about what underlies those types of pain. Chronic pain often involves persistent, low-grade inflammation. And neuropathic pain, which comes from damage or dysfunction of the nervous system itself, is very tightly linked to inflammation involving nerves and glial cells, and also altered nerve signaling.
Ah, so PEA's known mechanisms: reducing inflammation, calming glial cells like microglia, maybe modulating nerve activity, seem tailor-made for those conditions.
Exactly. It directly addresses the likely underlying biology. Beyond just general anti-inflammation, studies show PEA can specifically reduce hyperalgesia.
Hyperalgesia, that's increased sensitivity to pain, right? Where things hurt more than they should.
Precisely. PEA reduces this in various experimental pain models. One source suggests it does this, at least partly, by inhibiting a key inflammatory signaling pathway called NF-kappa, specifically within the dorsal root ganglia, or DRGs.
DRGs. Those are nerve cell clusters along the spine that relay pain signals.
That's right. So PEA might be calming down those relay stations. Another study looked at PEA for painful nerve damage caused by a chemotherapy drug, oxaliplatin. It found PEA might help by influencing pathways like NF-kappa B again, but also another one called NRF2, which is involved in the body's antioxidant defenses.
So it's not just reducing the inflammation around the nerves, but potentially directly calming the nerve cells themselves and maybe even boosting their own defenses.
That seems to be the implication from these findings, yes. The sources give examples like studies showing PEA reduced pain-like behaviors in mice after they were given formalin, which causes inflammation and nerve irritation, and that study where central administration of PEA reduced hyperalgesia.
Okay.
And importantly, when the sources discuss the human clinical trials for pain, they note that researchers don't just look at pain scores. They also evaluate whether people needed less pain medication and, crucially, how the pain was impacting their daily activities and quality of life.
That makes so much sense. For someone living with chronic pain, knowing if it helps them function better day to day is probably just as important, if not more so, than just a number on a pain scale.
Absolutely. It speaks to meaningful, real-world improvement. Now, another major area where the sources point to promising effects is neuroprotection.
Neuroprotection. Protecting nerve cells.
Yes, protecting them from damage or dysfunction.
Given everything we've just discussed about reducing neuroinflammation and potentially calming nerve activity, neuroprotection seems like a really logical next step, doesn't it?
It really does, because inflammation, especially involving those glial cells like microglia and astrocytes in the nervous system, is now understood to play a big role in the progression of many neurological conditions. Some of the reviews cited touch on this interplay. So, PEA's ability to reduce microglia activation, which we already covered, is inherently neuroprotective. But the sources mention other specific actions too, attenuating oxidative stress that helps protect nerve cells, and there's a really compelling detail from one study. PEA was shown to counteract something called reactive astrogliosis.
Astrogliosis. That's when astrocytes, another type of support cell in the brain, react badly to injury or disease.
Yeah.
Like forming scar tissue.
Sort of, yeah. It's a complex reaction that can be harmful in the long run. And this study found PEA could counter the astrogliosis triggered by beta amyloid peptide.
So beta amyloid. That's the stuff that makes up the plaques in Alzheimer's disease.
Exactly. So showing PEA can counteract a harmful cellular reaction induced by a key component of Alzheimer's pathology is.
Well, it's very interesting.
That is a very specific and potentially significant connection.
It is. And going further, one source mentions a specific formulation. Remember we talked about co-ultramicronized PEA. Sometimes it's combined with luteolin, a flavonoid, this combo called PELAT.
Some PELAT, okay. It was shown in one study to actually promote neuronal regeneration after spinal cord injury in an experimental model.
Promote regeneration. Not just protect, but help regrowth.
That's what that specific study suggested, yes. And looking at neurodegenerative conditions more broadly, the in-depth research request calls the preclinical studies in Alzheimer's models encouraging.
Okay, encouraging.
And it highlights, quite prominently, a phase 2 clinical trial. This trial used that specific co-ultramicronized PEA with luteolin formulation. They often call it co-ultrapelet. And in patients with frontotemporal dementia or FTD.
FTD, another serious neurodegenerative disease.
Yes. The trial results demonstrated potential in slowing disease progression.
Slowing progression in FTD, that would be huge.
It would be very significant, yes. And this specific formulation and this FTD trial are mentioned in multiple sources, suggesting it's a really active and important area of current research. They're clearly taking this formulation seriously for neurodegenerative conditions.
Okay, wow. So just to recap the big ones.
Yeah.
Robust evidence for chronic and neuropathic pain, potent anti-inflammatory effects across different systems, including the brain and gut, and really exciting potential for neuroprotection, maybe even slowing progression in conditions like FTD, especially with these newer formulations. That's quite a portfolio of effects.
It is a significant scope, definitely.
Are there any other areas the sources hint at, maybe where the research is a bit more preliminary or just emerging?
Yes, they do touch on a few other areas, though. As you say, the evidence is generally described as less developed compared to pain and inflammation.
Okay.
The in-depth research request guide mentions potential roles are being explored in certain mental health conditions. It specifically names depression and autism.
Depression and autism. That seems a bit of a leap from inflammation and pain at first glance.
Yeah.
How might PEA connect there?
Well, it likely ties back to that growing understanding of the brain-inflammation connection. There is a strong theme in some of the background sources, like that review by Scaper and colleagues, about how inflammation, either systemic inflammation throughout the body or neuroinflammation within the brain itself, is increasingly seen as a potential contributing factor to various neuropsychiatric disorders.
Inflammation, mental health link.
Right. So the thinking would be that PEA's documented anti-inflammatory and neuroprotective actions could potentially help mitigate this underlying inflammation, and by doing so, indirectly influence brain function, maybe affecting mood or behavior. But the sources we have don't really dive deep into the specific mechanisms proposed for depression or autism. They just mention it as an area of stress.
Got it. So a plausible connection via inflammation, but still early days in terms of research for those conditions. What else is sort of on the radar?
There's a brief mention of possible support for muscle health.
Muscle health.
And one source dives into its involvement in mitochondrial health, specifically in an experimental model of diet-induced obesity.
Mitochondria. The cell's power plants. How does PEA fit in there?
Well, this study, using obese mice fed a high-fat diet, found that PEA treatment seemed to help restore the balance of mitochondrial dynamics, how mitochondria change shape, divide, and fuse, which was disrupted in the liver of these obese mice.
Okay, so it helped normalize mitochondrial behavior.
Yes, and along with that, it also improved some related metabolic problems, like the levels of hormones involved in appetite and fat metabolism, such as leptin and adiponectin.
Huh. Connecting PEA to mitochondrial function, especially in the context of a metabolic disorder like obesity, that adds yet another interesting layer to its potential roles.
It certainly broadens the picture, and then we see more complexity emerging in PEA's relationship with the broader endocannabinoid system, beyond just that potential entourage effect we discussed. Well, one study cited suggests PEA might actually inhibit FAAH.
FAAH, that's the main enzyme that breaks down anandamide, right?
Exactly. So if PEA inhibits FAAH, that could lead to higher levels of anandamide. And in that specific study, they found this potential FAAH inhibition by PEA seemed to enhance anandamide's ability to slow the growth of human breast cancer cells in a lab dish.
Whoa! So PEA might indirectly boost anandamide levels, which then had an anti-cancer effect in that specific cell model.
That was the finding in that particular study, yes. Now, it's really important to stress none of these sources are suggesting PEA is like a standalone cancer treatment. Not at all.
Right, right.
Important caveat.
But these kinds of cell-level interactions are scientifically interesting. They hint at potential adjunctive roles, maybe supporting other therapies that researchers might be exploring in various cancer models like those breast cancer cells, or another study mentioned looking at a thyroid carcinoma model where an anandamide analog showed effects. It's about understanding the complex cellular signaling.
Understood. Basic science exploration of these pathways. Precisely. And one more related point. Another source notes that PEA, acting through PPAR alpha, might inhibit angiogenesis in inflammatory conditions like colitis.
Angiogenesis, that's the formation of new blood vessels.
Yes. And while new blood vessels are needed for healing, excessive or abnormal angiogenesis can actually fuel chronic inflammation and other diseases. This source suggested PEA helps curb this in colitis by reducing the release of VEGF.
VEGF, vascular endothelial growth factor, a key signal for blood vessel growth.
Right, so PEA acting via PPAR alpha might put the brakes on excessive blood vessel formation driven by inflammation in the gut, according to that finding.
Okay, so let's pull this section together. Beyond the core pain and inflammation story, we've got hints or emerging research related to mental health conditions like depression and autism, potentially via inflammation, support for muscle health, connections to mitochondrial function and metabolism in obesity models, complex interactions with the endocannabinoid system, possibly boosting anandamide by inhibiting its breakdown enzyme FAAH, and even potential anti-angiogenic effects in inflammatory states like colitis. It really does seem like PEA pops up or has effects in a surprising number of fundamental biological processes.
It really does. I think its nature as an endogenous molecule, something the body makes itself, combined with its ability to interact with key pathways like PPAR alpha and influence the balance of other related signaling lipids. It just makes it a subject of really broad scientific investigation across many different aspects of health and disease. And as we noted, the development and testing of specific formulations, like that co-ultramicronized PEA, sometimes with luteolin, the pelute or co-ultrapelute that was used in the FTD trial, that's clearly a focus. Although it's worth remembering that formulation was also tested in that study on smell problems after COVID.
Oh, right, the Carosmia study.
Yeah, and the results there were described as less clear-cut. It didn't seem to show a strong benefit compared to the control group, who actually started off with better smell function anyway.
So that kind of reinforces the idea that even with potentially optimized formulations, the effects aren't necessarily guaranteed across every single condition. The specific context matters.
Exactly. The specific biological situation and the patient population being studied make a huge difference. It's not a magic bullet.
Right. Okay, now shifting gears just a little bit. When we talk about natural compounds for things like pain and inflammation, other names inevitably come up. CBD cannabidiol is probably the most well-known example right now. How do the sources we're looking at position PEA compared to something like CBD? Is there any kind of direct comparison?
Yes, actually. That in-depth research request document specifically includes a comparative section, which is quite interesting.
Oh, good. What does it say?
It really highlights a key difference in their main proposed mechanisms of action based on the research reviewed. As we've spent a lot of time discussing, PEA's primary action described in these sources is that direct activation of PPAR alpha.
Right, the nuclear receptor, the genetic switch.
Exactly. And this is contrasted with CBD. According to the source's reference, like a review in the journal Neurotherapeutics, CBD seems to act through a much wider variety, a broader spectrum of receptors and pathways.
Okay, so CBD isn't focused on just one main target like PEA is with PPAR alpha.
That's the picture painted here. The sources mention CBD interacting with multiple different receptor types.
Like what? Can you give some examples?
Sure. They point to CBD hitting receptors like TRPV1, that pain and heat receptor we mentioned earlier, PPAR gamma, which is another member of the PPAR family, different from PA's main target, PPAR alpha. Serotonin receptors, specifically the 5-HT1A type, which is important for things like mood and anxiety. even mu and delta opioid receptors, the ones targeted by opioid pain medications, and another receptor called GPR55, which has various roles.
Wow, that's a lot of different targets. TRPV1, PPAR, gamma, serotonin, opioid receptors, GPR55.
It is. And that review also notes CBD has other effects that seem independent of the classical CB1 and CB2 cannabinoid receptors, plus it has significant antioxidant capacity on its own.
So if I'm trying to
Yeah.
simplify this, PEA's story, according to these sources, is largely about activating one key genetic switch, PPAR alpha, whereas CBD is more like a multi-tool, interacting with a whole diverse set of receptors and pathways involved in pain, inflammation, mood, and more.
That's a pretty good way to summarize the distinction drawn by these sources regarding their main reported mechanisms for these kinds of effects. It's a difference in specificity versus breadth of action, potentially.
Okay. And does this comparison extend to the actual evidence for effectiveness?
It does. The in-depth research request makes a specific point about the clinical evidence for pain. It states that the existing meta-analyses, those studies that pool data from multiple clinical trials, seem to provide, and again I'm quoting, more consistent support for PEA's analgesic effects compared to CBD's.
More consistent support from meta-analyses. That sounds pretty significant. It suggests that, at least based on the clinical trial data summarized so far in these reviews, the evidence pointing towards PE actually working for pain is perhaps clearer or more reliable than the evidence currently available for CBD,
That's the interpretation presented in this particular set of sources, yes.
based on their reading of the meta-analytical literature for pain relief.
Interesting. What about safety? Is that part of the comparison? Yes. Safety is another point raised. The sources, as we've noted, consistently emphasize PEA's strong safety record, and they partly attribute this to the fact that it's endogenous, your body makes it already, and its primary action is through these well-characterized pathways, mainly PPAR alpha activation.
Right.
So the source doing the comparison concludes that this overall profile, the potentially more consistent evidence for pain from meta-analyses, the strong safety record, the well-defined mechanism, quote, may position it as a more straightforward and potentially favorable option compared to CBD for certain individuals seeking relief from pain and inflammation. That's based on the balance of evidence presented in these specific documents.
Okay, that's a really clear framing. It's not saying PEA is definitively better overall, but suggests that for pain and inflammation, based on the current summarized evidence for efficacy, combined with its safety profile and mechanism, PEA could be seen as a potentially more straightforward or favorable choice for some people compared to CBD.
Exactly. It's highlighting those specific points of comparison, the nature of the evidence from meta-analyses for pain, the perceived safety profile, and the primary mechanism of action. Those seem to be the key differentiators discussed in these sources.
Got it. Okay, this is all incredibly informative. Now, if someone is listening to this and finding it intriguing, maybe thinking about this in a practical sense, what do the sources actually say about how PEA has been used in the studies? Like, what kind of dosages are common?
Right, practicalities. The sources do provide some general dosage information pulled from the research papers and clinical practices they reviewed. For adults taking PEA orally, by mouth, a common range reported in many studies is between 300 milligrams and 1,200 milligrams per day.
300 to 1,200 milligrams a day.
And typically, that total daily amount is divided up, often taken in 2 or 3 separate doses throughout the day.
Splitting the dose, that makes sense. Probably helps maintain more consistent levels in the body.
That's usually the rationale, yes. Keep levels steadier. Now, some sources and clinical reports do mention that daily doses have gone higher, even up to 2,400 milligrams per day, and these higher doses also appeared to be safe and effective in some contexts. But that 300 to 1,200 milligrammer range seems to be the most common window studied.
Can you give a little more context? Like, what doses were used in those pain studies versus, say, the FTD trial?
Sure. So those meta-analyses looking at chronic pain trials, they often included studies using dosages right in that main 300 to 1,200 milligram per day range.
Right.
The specific FTD clinical trial we discussed, the one using that special co-ultrapellet formulation, that one used a total of 1,400 milligrams of PEA per day, but it was split into 2 doses of 700 milligrams each, taken twice daily.
1,400 milligrams a day for the FTD trial.
Got it. And then studies looking at muscle health tended to use doses more on the lower end of the scale, often around 300 to 350 milligrams per day.
So the dose might depend quite a bit on what condition is being studied or targeted.
It seems that way, yes. Dosage often needs tailoring.
And how long did people typically take it for in these research studies? Is it usually a short-term thing or longer?
In the formal research studies, the duration often ranges somewhere between 2 weeks and 12 months, so a few weeks to about 3 months.
Okay.
However, the sources do acknowledge that in real-world clinical practice, people might use it for longer periods. And some of the trials themselves went longer, like that FTD trial, which ran for 24 weeks, about 6 months. So longer durations are certainly studied and used.
What about for kids? Is there any information on that?
The information is definitely less extensive for children, but the sources do mention that dosages around 600 milligrams per day, taken for up to 3 months, have been used in studies involving children and adolescents, specifically in the 4 to 17-year-old age range.
Okay, good to know there's at least some data there. Now, are there ways to use PEA other than just swallowing a pill or capsule?
Yes, PEA is also formulated for topical application, so you can find it in things like creams, lotions, even some eye drops contain PEA.
Topical PEA?
Yeah.
Interesting. What do they say about the safety of using it on the skin?
The source is generally considered topical PEA to be possibly safe, based on the available data, specifically for use up to about 28 days. The data for longer-term topical use seems more limited in these sources.
Gotcha. And circling back to overall safety, you mentioned its strong safety record several times.
Mhm.
Can you elaborate a bit? What does the research generally say about how well people tolerate it? Any common side effects mentioned?
Yeah, the sources are pretty consistent on this. They repeatedly highlight PEA's favorable safety profile and that it seems to be generally well tolerated. As we discussed, its endogenous nature, the fact that your body makes it, is often cited as a contributing factor to this good profile.
Right.
Now, they are careful to point out that, like with any compound, more extensive clinical studies across diverse populations are always helpful to fully map out any potential rare side effects or interactions. That's standard scientific caution. But based on the existing research reviewed in these documents, the overall picture presented is one of minimal adverse effects and good safety, especially when you compare it to some other medications or compounds used for similar things like chronic pain. They don't really detail specific common side effects, suggesting they might be infrequent or mild, but the general consensus from these sources is definitely positive regarding safety and tolerability.
That's really important context for anyone listening and trying to understand the full picture. So it appears to be well tolerated in the studies conducted so far, based on this information.
Based on these sources, yes, that certainly seems to be the conclusion they draw.
Okay, let's try to zoom out a bit now and connect some of these dots. We talked earlier about broader concepts mentioned in the sources, like neuroinflammation and the specific roles of cells like microglia and mast cells. How does PEA's story, especially its action through PPAR alpha, really fit into this bigger picture of how cells communicate and respond in the body?
Right, this is where it gets really interesting, seeing how the mechanism fits the bigger biological context. Those reviews, like the one by Scaper and colleagues, really emphasize how critical cells like microglia and mast cells are not just in neuroinflammation, but in inflammatory processes almost everywhere in the body.
Yeah, they seem to be key players.
They are. And PEA's demonstrated ability, highlighted in these sources, to directly reduce the activation of microglia and inhibit the degranulation of mast cells. Well, that directly targets these key players. Like PEA is intervening right at the level of the cells, orchestrating the inflammatory response.
So it's influencing the conductors of the inflammation orchestra, so to speak.
That's a nice way to put it. And this applies whether the inflammation is happening in the nervous system or out in peripheral tissues like the gut, where we saw that specific study linking PEA's effects to those enteric glial cells, too.
Ah, yes, the gut's own support cells.
Exactly. So it seems capable of modulating the behavior of multiple distinct cell types that are central to inflammation and tissue response in different locations.
It's not just a broad, non-specific effect then. It seems targeted at these crucial cell types.
Precisely. And then you have its core interaction with PPAR alpha. That connects PEA directly to this incredibly important system where lipid signaling molecules, fats, and related compounds directly influence gene expression.
Right, the genetic switchboard.
Exactly. It's
Yeah.
a fundamental and really powerful way the body controls everything from basic cellular functions and metabolism to how it mounts a defense, how it responds to stress, injury, or infection. PEA is tapping right into that control system.
And what about its connection to the endocannabinoid system? How does that fit in the bigger picture?
Even though it's generally not considered a classical cannabinoid receptor agonist, it doesn't directly hit CB1 or CB2 strongly, its presence, and proposed interactions within that broader endocannabinoid network add another layer. Think of it as a modulator within these internal signaling systems that are all about maintaining balance, homeostasis, and coordinating responses to challenges. It seems to be acting both upstream, influencing genetic programming via PPAR alpha, and also downstream, affecting the immediate behavior of specific cells like mast cells and microglia involved in defense and repair.
That really paints a picture, doesn't it? A molecule made by the body, involved in very fundamental ways. The body maintains health, controls inflammation, and responds to various kinds of stress or injury.
It does. It strongly suggests PEA is an integral component of the body's own sophisticated intrinsic system for managing inflammation and promoting resilience and protection, right down at the cellular level.
Okay, so let's bring this deep dive toward a close. We've covered a lot of ground on palmitoylethanolamide, or PEA. Based on this whole stack of sources we've explored, what are the main takeaways? What does this all mean?
Well, first and foremost, we've learned PEA is a natural lipid molecule, something your own body produces to act as an internal signal.
Right. And its main way of working, according to all this research, seems to be by activating that PPAR alpha receptor inside the cell nucleus. That acts like a genetic switch, influencing genes involved in calming inflammation and protecting cells.
And the research bundle we looked at provides pretty robust evidence, especially from meta-analyses supporting PEA's potential use for chronic pain and particularly neuropathic pain. There are also really promising findings for reducing inflammation across different tissues, with significant effects shown in models of nervous system inflammation and also in the gut.
And beyond those main areas, the sources definitely indicate potential in related fields. Things like neuroprotection, maybe even helping support nervous system recovery and influencing mitochondrial health. There's ongoing research exploring these links, including those specific formulations, like the co-ultramicronized PEA with luteolin, that have been tested in clinical trials, like the one for FTD.
And finally, that comparison point is interesting. When placed alongside a compound like CBD for pain and inflammation, these specific sources suggest that the evidence from meta-analyses might currently offer more consistent support for PEA's pain-relieving effects. That, combined with its strong safety record and reliance on these well-defined biological pathways like PPO-alpha, positions it as a potentially favorable option in some cases.
It really is a fascinating example, isn't it? How digging into the science of these natural endogenous molecules our body already uses, and understanding their precise mechanisms, like how PEA interacts with PPAR alpha and influences key cells like mast cells, microglia, and even those enteroclia, how all that detail can point towards new and potentially quite powerful ways to support our health and bolster the body's own protective and healing processes.
Absolutely. I think PEA's ability to modulate these really critical inflammatory and support cell types, primarily through that PPAR alpha pathway, clearly establishes it as a molecule with significant therapeutic potential. And it's obvious the scientific community is actively and broadly investigating that potential across a whole range of different conditions.
It really makes you wonder, given that PEA is involved in modulating the body's fundamental inflammatory and protective responses, and it does this largely through a nuclear receptor like PPAR alpha, a receptor that directly dials gene expression up or down in various tissues, what other, maybe completely unexpected, wide-ranging effects or connections might further research into this natural molecule uncovered down the road? Things we haven't even begun to imagine yet.
That is a truly fascinating question, isn't it? Exploring the full depth and reach of these endogenous signaling systems, it really feels like we're still just scratching the surface, an ongoing journey of discovery.